welcome everyone to Osteobites. My name is Christina Iptoma, and I am mom to Osteo Angel Dylan and Director of Scientific Programs at MIB Agents. And today on Osteobites, we are talking with Dr. Lopez Fuentes. She's a postdoc at UCSF in the Sweet-Cordero Lab, which is an osteosarcoma research powerhouse. And she will be discussing her work studying two epigenetically distinct cellular states in osteosarcoma that are regulated by a state-specific set of transcription factors driving differential drug response. Thank you so much, Dr. Lopez Fuentes, for joining us today on Osteobites. We're thrilled to have you. And thank you also to Walker for joining us as our panelist today. Walker is an osteo warrior, and he's also a member of our MIB Agents Junior Advisory Board. So I wanted to start first with a little bit more about our guest today. A postdoc at UCSF, Dr. Lopez Fuentes completed her master's degree in biochemistry and molecular biology at the National University in Mexico. She then completed her PhD in molecular biology at a research center in Mexico called IPICYT. And during her postdoctoral training, she has continued to build on her previous work in epigenetics by addressing questions regarding the epigenetic regulation of pediatric sarcomas at the Sweet-Cordero Lab at UCSF. And her research focus is on defining the mechanisms of epigenetic gene regulation in osteosarcoma. Dr. Lopez Fuentes is also a 2022 Outsmarting Osteosarcoma Young Investigator Because of Charlotte grant recipient, and we are so very excited to welcome her to Osteobites today to share her work. So thanks so much for joining us, and welcome to everyone else joining our call today. And please feel free, if you have any questions for Dr. Lopez Fuentes, just feel free to pop them in the chat. And before we get started, I have a few other announcements because we've got lots going on. Save the date and mark your calendars for Factor 2023. It's our annual conference. We are going to be in Atlanta this year, June 22nd to 24th. Reg will open in February, and I'll put a link in the chat with some more info about that. We are accepting abstracts now for Factor. The deadline is Feb 24th. And I will also pop a link in the chat. You can get more information on our website. And there is still time to apply for our Outsmarting Osteosarcoma grant. Applications are due Feb 3rd, so I think we're almost at like the week countdown. And we do have $100K annual awards, as well as $50K one-year grants for young investigators, which is what Dr. Lopez Fuentes received last year. And as we did last year, we do expect to fund multiple awards again this year. Last year, we were able to support 6 PIs. And so we do encourage you to apply. You can apply online at Proposal Central and find more information on our website. And last, to stay on top of all these programs and events, please subscribe to our monthly newsletter, Connective Issue. Lots of information of interest in there. And to make sure you get it in your inbox every month, just go to our website and click on the join button, and you'll get added to the list to get that. So Walker, I'm going to hand it over to you if you wouldn't mind introducing yourself.
Yeah, hi, my name is Walker Smallwood. I'm an osteo warrior. I'm the part of this year's Junior Advisory Board. And Dr. Lopez, you can take it from there.
Thanks, Christina, for this kind invitation. And also, I want to thank this amazing foundation at the MIB. And also to my sponsors, because of Charlotte, because this allows to keep going with this project. Today, I'm going to talk about my project during my postdoc at UCSF with Alejandro Sweet-Cordero. And first, I want to start with some concepts about epigenetics. So, every cell in our body contains the same DNA. Yet somehow the DNA provides the basis for each cell's individuality. And this can be explained by epigenetics, which literally means above the genes. You can see here in this figure how the DNA is organized in different chromosomes territories in the nucleus, but also the DNA is wrapped around these proteins that we call nucleosomes. And also these nucleosomes have different modifications that we call histone modifications that allows two different states. The on state or open state you can see here how it's a relaxed conformation where the genes encoded in the DNA can be transcribed. And this off conformation when the chromatin is really condensed, and usually these genes are not transcribed, and we call this off. Epigenetics plays a role in different cancers, and because can turn off a tumor suppressor, but also can turn on a tumor helper. These epigenetic modifications do not change the DNA sequence, but instead affect how cells read the genes. So in my project, we're focusing this on state or open chromatin. We're analyzing what DNA is accessible inside a cell can tell us what the cell is capable of doing at a given time. And using this information, we can know what genomic regions are open and what are closed, and we can define a cell state. And by defining a cell state, we might predict drug response. We are using a protocol that is called ATAC-seq that, in a nutshell, quantifies DNA accessibility. So in this figure, you have the off state here to the left and the on state here to the right when the chromatin is relaxed, and you can see a gene that is transcribed. And we are using an enzyme that is a transposase that basically is coding only the open genomic regions. So you ended up with this DNA that were cut around the nucleosome, and after a DNA purification, you can sequence the fragments and these sequencing peaks corresponding to open chromatin.
Dr. Lopez Fuentes? Yes. All right, can I ask just a clarifying question? So when you're talking about epigenetics, like, a lot of times we talk about mutations and, things like translocations or things like that. So is this like a subcategory of that, or is this completely separate where it's not anything to do with gene mutations, but it's how the cells are reading them, like if they're turning things on or off? Like where does that kind of go in the hierarchy of things versus like when we talk a lot about mutations in genes?
It will be an upper level, because when you are looking at open chromatin, you also will have a lot of intergenic regions, but also regions where the genes are encoded. What we can do to address these mutations on translocation that is pretty common in osteosarcoma is do a correction by copy number. After you have all this data, you can correct by copy number and then use the data after this normalization.
Okay, so if I understand it correctly, epigenetics is kind of one of the things that results in some of those different mutations and translocations.
Yeah, what this, for example, if we go back to this slide, when you are talking about epigenetic modifications, you are talking about that these histone tails are marked with acetylations, for example, or methylation that will tell the cell to keep our open region. But this is the same DNA, right? The mutations and translocation will be in the DNA, and the DNA will be wrapped around these nucleosomes. But these conformations between on and off is like they can go back and forth in the cells. So sometimes you can have a translocation that is off, or sometimes you can have a translocation that is on.
Okay, got it. Thank you.
So if we keep going here, in this project, we have 3 different goals. The first one is define these epigenetic cell states in osteosarcoma, which means define the open genomic regions in osteosarcoma tumors. And the second goal is define the markers to predict these epigenetic cell states in patient samples. And the third one, or probably the most important, we want to validate drug response in vitro and in vivo after defining the cell states. We are using a set of 45 samples. We are doing 2 replicates per sample. That means 2 libraries per sample and different type of samples. We are starting with patient samples that can be primary samples or metastatic samples. And then using a piece of these patient samples, we implanted in a mouse, and we call that patient-derived xenograft or PDX. And from that, we can generate cell lines that we will call PDX cell line. So in this set of samples, we will have also the commercially available cell lines, that there are no PDX cell lines, there are only cell lines and have been available for decades in osteosarcomas. So using all these samples and doing a tag-seq, that is this protocol to know what are the open genomic regions. After we analyze all this data, I'm showing here a PCA, which is a condensed form to show this data in 2 dimensions. And in pink, you can see the commercially available cell lines, and in blue, you can see the PDX cell lines. And you can see how there is a lot of heterogeneity when we are talking about chromatin accessibility in osteosarcoma. I want to point out that the commercially available cell lines are clustered together in this corner, and the PDX cell lines are across the plot, which is suggesting that we have more heterogeneity between the PDX cell lines than the commercially available cell lines. Then when we include the PDX, which are the patient-derived xenografts that were in mouse, these PDX never touched the plastic. They were using after we extracted the tumor from the mouse, it was cell suspension was generated, and then the library was done. And you can see here how some of the PDX set are clustered here to the left and is forming another smaller cluster here. And also some of the PDXs are clustered here to the right. Then after we included the patient samples, for the patient samples, each sample is a biological replicate, right? Because we are taking a different piece of the tumor each time that we generate a library. Most of the patient samples are clustered here to the left, and only 1 patient sample is clustered here to the right. Using this first dimension that you can see here in the x-axis, we are defining at least 2 different main clusters using this chromatin accessibility data. We call this cluster here in green cluster 1 and this one in orange cluster 2. And then we asked...
Could I ask a question right there? And even on the previous slide. So, because these are the patient samples, then the PDX and the PDX, the cell lines, they kind of mount together, right? Like you have some samples where they're all 3. And so do you see that in general, they are kind of plotting in the same area? Like I see P708, the patient and the PDX are kind of in the same.
Yeah.
Yeah. Did you basically see that the PDX is similar to the patient sample and the...
We have, usually the patient and the PDX are in the same cluster. But we have some examples where we have the PDX in one cluster and the PDX cell line in another cluster, which is suggesting how these tumor cells have a lot of plasticity, and probably they can go back and forth between these 2 cell states. Yes. And we are exploring that hypothesis using single cell data. That I have a small figure at the end. That's a great question, actually.
Thanks.
And then we ask, what are the genomic regions that are different between these 2 clusters, right? And for that, what we did was a differential chromatin accessibility analysis, where you can see here that each row is 1 genomic region. And for example, red means that these are open regions. We have around 1,500 genomic regions that are open in cluster 2 here, and there are closed in cluster 1, and the opposite. We have around 2,400 genomic regions that are open in cluster 1 and closed in cluster 2. Using this differential analysis, we can do the assignment of these genomic regions to genes, right? Because when you have genomic regions, it's difficult to interpret the data. But if you do the assignment from genomic regions to genes, it's easier to assign this gene to different pathways. This is just a snapshot showing how the differential analysis look like. This is a genomic region of chromosome 6. And you can see, for example, here in orange, that this part of the genome is open in this cluster, but it's not in cluster 1, and the opposite, right? This is a good example how this genomic region is open in cluster 1, but it's not in cluster 2. So we are using GRADE to do the assignment to these genomic regions to genes, and using a window around 1 megabase, and then we ask what are the enriched pathways per cluster. And we found that for cluster 1, we have genes related to bone mineralization, osteoblast differentiation, cell development, mesenchymal cell proliferation. And for example, for cluster 2, we saw pathways such as regulation of cell migration, cell substrate adhesion, morphogenesis, regulation to cell communication. This type of analysis can give us an idea that what is happening in these two different cell states. And then we ask what drives the existence of these two clusters. Using the same assay, ATAC-seq, you can also answer the question, what are the transcription factors that are bind to the DNA of these osteosarcoma tumor cells? Because the transcription factor that is bind to the DNA is protecting the DNA from the transposase, the enzyme that is coding the open regions. So the enzyme will cut around the transcription factor, right? In this case, this blue is the transcription factors, and this one is the enzyme that is coding. So the transcription factor is protecting this region of DNA from the enzyme, right? So you will have this part of the DNA will be sequenced because we will cut around of the transcription factor. And using this rationale, we ask what are the transcription factors that are in these two clusters, because these transcription factors are the ones that are orchestrating the gene expression in these two different cell states. And we found that for cluster 1, we have RUNX3, RUNX2, MEOX, MEF2B, MEF2A. And interestingly, these transcription factors are involved in development. Most of these transcription factors have homeoboxes. And for cluster 2, we found that most of the AB1 complex members, such as FOSL1, FOSL2, JUNB, but it's still not clear what is the role of the AB1 complex. It has been described that AB1 complex has many roles in cancers, but we don't know what is the role in this epigenetic cell state yet. And then using the PDX cell lines and some of the commercially available cell lines, we ask if we can see enriched protein expression of these transcription factors and other proteins between these two clusters. So we do immunoblottings, and for example, here RUNX2, which is a cluster 1 transcription factor, you can see how it's enriched in the PDX lines that are cluster 1, and is not in cluster 2. And the same is happening for another transcription factor, such as FOSL1 or FOSL2. We do see higher protein expression in cluster 2 because these two transcription factors, we found them in cluster 2, that specific cluster 2. And we did this using total lysets, but also we did the fractions, the protein fraction, the cytosolic, the nuclear, and the chromatin fraction, to ask if we can see this enrichment just in the chromatin fraction, suggesting that these transcription factors can be master regulators of these two different cell states. And actually, we do see it. Fossil 1 and fossil 2 are enriched in the chromatin fraction on the cell lines that are cluster 2, and RONGS 2 and RONGS 3 are enriched in the chromatin fraction of the cell lines that are from cluster 1, which was really great that we can have this validation of the in silico analysis.
And is it that completely distinct? Like there was another, like, is there any overlap, or are there any proteins where you're seeing them in both clusters? Is it that clean?
Yeah, exactly. It's not perfect. For example, we found FOS here in this. You can see how FOS is not, at least in the protein expression, is not cluster specific. You can see that some cell lines have enrichment, but and is not cluster dependent. Yes. But the fact that we can see enrichment in the chromatin fraction is suggesting like we can have these pioneer transcription factors that are opening the chromatin and regulating the genes in these open genomic regions. And then because we can have, after the peaks assignment to genes, we can have a list of genes from the chromatin accessibility assay. We decided to do the intersection using the gene expression data, right? Because you can think of this, you can have the expressed genes that are in the open genomic regions. So when we do the integration, we use only RNA-seq data, and we do the assignment to the epigenetic clusters, cluster 1 and cluster 2. This is PCA. You can see dimension 1 and dimension 2, and this is just color-coded by the ataxic clusters. In orange, you can have cluster 1, and in green, you can have cluster 2. And it's also interesting that you don't see this separation between these two clusters using gene expression, the way that we see it when we do the same PCA analysis using chromatin accessibility data. And then we decided to do a differential analysis between these two clusters to have a list of genes that are expressed in a cluster-specific manner. And we ended up with around 900 genes after this differential analysis using only gene expression data. And then for the integration, we started with the genes that were from the peaks from the ataxic analysis that are unique per cluster. And then we included the differential analysis using only gene expression data. We sort out the genes using only the cluster 1 specific genes and the cluster 2 specific genes, and do an overlap between these two different differential analyses, right? And we ended up with a gene signature of around 300 genes. That in this gene signature, we have expressed genes per cluster that are in open genomic regions per cluster. And using this gene signature, we take all the patient samples that we have gene expression data. This heat map was done using only patient samples because for the ataxic, unfortunately, we didn't have a lot of patient samples to do chromatin accessibility as assays. But for gene expression, we do have a good set of around 50 samples. So these are around the 50 samples that we have gene expression data, and every row is the gene signature that we defined, the 300 genes that we previously defined doing the integration. And the question here if that we can see these two clusters using just human data. And actually, we do see it. This is just a preliminary result, but it's really promising to see that using the gene signature that was defined previously, we can see these two clusters. For example, this one will be cluster 2, and this one in green will be cluster 1. And then after the assignment of these patient samples to one cluster, we ask if we can see an impact in patient survival. We use only primary samples. We remove the metastatic samples to do the survival analysis. And we saw that cluster 1 shows a lower survival probability than cluster 2. We are redoing this analysis in a more robust way with a collaboration that we have on UCSC, Santa Cruz. And we will have this analysis in a more robust way and probably including more samples.
Dr. Lopez Fuentes, I'm curious if you were to include metastatic samples, can you explain to us first of all why you were looking only at primary samples? Is it just more for like predictive or?
Because when you are doing a survival analysis, the rationale was that if you already have a metastatic sample, the survival decrease like a lot than a primary samples. And also because we have at some point primary samples from the same patient and then a metastatic sample. So we will have two samples from the same patient. So if we only use primary samples, we are using one sample per patient.
Right. And I'm curious if you were to look, because you have this nice, because you have the same samples for the same patient, is it at all possible that it would change in a metastatic sample? Could they move from one cluster into another cluster?
Yes. We have a few examples actually. This is not in this heat map, but we have that analysis where it's not always the case, but we have seen this, that we have primary samples in one cluster and then the metastatic samples are in the other cluster. Yes. Then the next question that we have was if we can define specific markers to classify patient samples into this epigenetic cluster, because this will be like the first step to translate this work to the clinic, right? Because probably it's not easy to do just a tag-seq for all the patient samples. It will be easier to have just markers that you can use to classify these patient samples. We tested some of the genes from the signature that we described, the 300 genes, and we found that SATB2 and SP7 are really good markers for cluster 1. And I also want to point out here that for this validation, we are using PDX that were grown in an orthotopic model. So that means that the tumor was grew in the leg of a mouse, and the orthotopic model is mimicking in a better way the disease than the subcutaneous model. So it was great that this worked out, that we can see an enrichment of these markers in these tumors that were cluster 1. Example, you can see here two tumors that were growing with PDX lines from cluster 1, and you can see the quantification at the bottom for SATB2 and SP7. These two genes are involved in ossification, but also bone formation. For the next cluster, we found FRA2, which is the protein, and FOSL2 is the gene. And interestingly, we do have FOSL2 as a transcription factor that is cluster 2 specific, but it's also working really good as a marker in these PDX tumors. And you can see the enrichment, the quantification here to the right, and this is just the histology of these tumors. we can also ask if these epigenetic cell states can be associated with different cell types. In order to answer that, we use ataxic data from this paper, which is a non-cancer paper, but in this paper they describe human skeletal stem cells, and we call this HSSC, and they use different markers to describe this population. And using this data from this paper as stem cells, but it's human skeletal stem cells, and also osteoblast cell line. We did the ataxic on this cell line that is also a no-cancer cell line. And we found that when we include these two data in our set, the human skeletal stem cells are aggregated here with our cluster 1, and the osteoblasts are aggregated here in our cluster 2. This is suggesting that the level of differentiation for cluster 1 is, I will rephrase this, the cluster 1 is less differentiated than the cluster 2. And we are exploring further this possibility using the single cell data, RNA-seq data. And then for our next goal was to see if we can find differential drug response between these two clusters, right? Because it's great that we have all this data and we are gaining insight on how is the behavior of these two clusters. But what we really want to know is if we can find this differential drug response after the classification in these two cell states. We are using an orthogonal data set that was generated in our lab by Lian Sales. And you can see the panel of drugs and targets that were using that were used in this data set is across 40 different targeted agents. And they were using a smaller panel of the PDX cell lines. So after we classify these PDX cell lines, that we also have ataxic data and we know exactly what is the cluster that they belong to, we ask if we can see differential drug response between clusters using this panel of this data set. And we do find differential drug response actually we saw that cluster 2 is responding to a MEK inhibitor called trametinib, and cluster 1 is responding to aurora kinase B inhibitor and aurora kinase A inhibitor. This drug response was generated in an in vitro system, right? The cell lines were growing on a plate and then the drug was added and then test what is the inhibition of these cell lines. And then in order to validate the differential drug response, we are using in vivo models. In this case, it's a subcutaneous model. And we started with a cluster 1 PDX cell line. And in our hypothesis in the in vitro model, we saw that cluster 1 responds to aurora kinase B inhibitor, and it's the same that is happening in vivo. You can see here in blue how the vehicle is really growing fast, but when the tumors were treated with aurora kinase B inhibitor, the tumors are basically not growing. And actually, some of the tumors are shrinking after 24 days of treatment. And you can see here to the right how the difference is significant. And also, interestingly, the tumors are not responding to trametinib, which is the drug that is cluster 2 specific. And then we do the opposite experiment using a cluster 2 PDX cell line, which in vitro is responding to trametinib. And we do see a response of trametinib. However, the tumors didn't shrink. It's only the tumors are growing slower when we are using this subcutaneous model with this PDX cell line. And then you have to think of this data as this is bulk data. So what we're really seeing is that one cluster is predominant in these tumor cells, right? But there is also the possibility that you can have these two cell states in one tumor. So we are going in that direction in this project, because when we evaluate another PDX line that is cluster 2, you can see here the top left how this line is cluster 2, and the hypothesis is that cluster 2 is responding to trametinib. When we do the in vitro assay, the cell line didn't respond to trametinib, but it does respond to aurora kinase B inhibitor, which is a cluster 1 inhibitor. However, when we do the in vivo data, or the in vivo experiment, we saw how in a subcutaneous tumor, the cell line is responding to trametinib. So that this is suggesting that we probably have the two cell states in one tumor, and the cells have a lot of plasticity, and they probably can go back and forth, depends on the environment, which is really interesting. And you can see the quantification here to the right. And then we tested the PDX from the same patient. This is the same number, 833. And using the combination to test if we can target both cell states with these two drugs. And you can see here the plot over time, how the vehicle is growing really fast. And then the single drug, which is trametinib, the tumors are growing slower. But when we treat with aurora kinase B inhibitor, because this PDX is cluster 1, the tumors are responding better. But when we do the combination, the tumors are responding even better. And you can see here how with the combination, the tumors are not growing. Most of the tumors are shrinking in this, you can see here in this plot, how this is the negative scale here. And also between the single drug and the combination, the difference is significant.
Wow, so that was one PDX. Were there other examples where it was a similar?
No, we only have this example for this hypothesis, and everything started because we saw that the cell line wasn't responding to the drug supposed to be responding. So we went further and do the in vivo for the cell line and then the PDX. Because when you do these kind of experiments using a PDX, has to be really well planned, right? Because you have to collect the tumor from a mouse and then do a single cell suspension, and in the same day has to be implanted in the set of mice. So we don't have a lot of PDX growing to this experiment. Usually we regrow a PDX, but just to be stored, right? Because we have cryovials of this single cell suspension in liquid nitrogen, and then, but this experiment was really from a fresh tumor that we harvest the mouse, the tumor cells from the mouse the same day. But yeah, we are planning in doing more experiments to see if the combination is a better option than the single drug.
Yeah, that's really interesting.
And then in summary, we are describing two epigenetic clusters in osteosarcoma with a distinct transcription factor sets that are orchestrating gene expression. And we saw that these two epigenetic clusters are showing differential drug response. And for our next steps, we are evaluating if these two clusters are driven by enhancer regulation. And to answer that, we're doing libraries of different histone modification, that was we were talking at the beginning. And for example, this 27 acetylation is a transcription activation mark, but it's also an enhancer mark. And this is just a preliminary result, but you can see here how the histone mark is in different genomic regions per cluster. You can see each row is one genomic region, and this is for one cluster here in blue, and this is for the other cluster here in orange. And then for another, the next step in this project is to evaluate what is the proportion of the cells that are cluster 1-like or cluster 2-like. And this is just an example how, for example, this cell line, this is single cell RNA-seq, and you can see here in green how you have cells that are cluster 1-like and you have cells that are cluster 1-2 in this PDX cell line. And the opposite, right? For this cell line, you have cells, each dot is a cell that are cluster 2-like, but also you have some cells that are cluster 1-like.
So that's really neat. So with the single cell sequencing, you could ideally for each of these samples, well, actually, I don't know how much work it takes to single cell sequence each sample, but ideally you could then kind of start to break out for each sample the proportion, right? And kind of then evaluate further kind of what you just did, like response.
Yes, exactly. This is single cell RNA-seq. The idea would be to do single cell ATAC-seq. Yeah, we are thinking about that, but it's a really expensive experiment, and probably we cannot do all the 45 samples using this single cell technology. And then I would like to thank Alejandro and our lab, which is an amazing lab to be doing this type of science. Everyone is willing to help. And a special thanks to Lian, Maria, Andrew, Alex, and Elizabeth. And this is and our collaborators, and for sure the funding, the MIB, UCMexus, CONACYT. And this is my email. If you have more questions that you don't want to ask here, you can email me and I will be happy to answer them.
Great. Thank you so much, Dr. Lopez Fuentes. We're going to open it up for questions. If anyone has any questions, please feel free to pop them into the Q&A. Walker, did you have any questions you wanted to?
Yeah, I have one question. So you were saying how the cells kind of have neuroplasticity and change back and forth based on their environment. So this, you might have already answered this, but do you guys use both drugs at the same time? Or like why would you not just do that no matter what, since it seems like the cells can change back and forth?
Yeah. At the beginning, the idea was to have this differential drug response because we want to look if the cluster 1 samples are responding better to one drug than another, right? But then we realized that both cell states can be in the same tumor, which makes sense, right? Because there is a lot of heterogeneity in these osteosarcoma tumor cells. And yeah, you're right. Probably the strategy will be to have to treat the tumors with both drugs instead of one.
Gotcha. That makes sense. What would be like the timeline for this to go to like human trials?
That's a hard question. I don't know the logistics about how to jump from these type of projects. I will say that you have to get a company involved because I know that a clinical trial is really expensive for a academic research lab to do.
No, that makes sense for sure.
Yeah, but that would be the next step for the translation of this word, to do a clinical trial after the classification of patient samples in these two clusters. Yeah.
And then in terms of translating that to the clinic, I mean, you had talked about the different biomarkers. And so, like practical, like if you were to see, like in the dream world, as this progresses, and like how would you kind of see that clinical application of testing, like being like patients being tested for whether or not, and it is a little complicated because since, as you said, like they can have cluster 1 and cluster 2 within the same tumor, and then that also maybe evolves too as they with current disease. And so, how often would you basically have to, check and see kind of which cluster you're in at that particular time? It just seems like there are a lot of variables. So I'm just curious, like how you would see this progressing to identifying patients that would respond to a
Yeah, we are working on that for the markers per cluster. I will say that the first step will be to test. First, do the validation using a set of samples that we know what is the cluster they belong to with a TMA, right? Because it will be faster to have around, I don't know, more than 20 samples in one slide and just test different antibodies. And then do probably the same, but with patients, and see if we can identify these two markers in these patients. But we have to have the ataxic data to do this validation of patient samples. We are struggling with the number of patient samples that we have. Actually, we will want to have more patient samples to do this validation, and we are working on that too.
And both of the drugs that you mentioned, Dr. Lopez Fuentes, those are both readily available drugs? Are they?
No, trametinib, I know that is FDA approved, but autokinase B is not.
Got it. So there's some work there too to have a drug that's available.
I think there are clinical trials evaluating the toxicity at the moment for the autokinase B inhibitor. Yeah.
Great.
So compared to like a chemotherapy where it kind of attacks like many of the cells in your body, do these drugs, like being tumor suppressors, do they solely attack like that tumor and that type of like cell that they go after, or do they go after more or like other cells in the body as well?
That's a great question. I don't know the answer, but I will say that it will attack other cells too, because when you are treating the mice with these drugs, the mice are, in general, they are doing well, but at some point, if you keep going with the treatment, they will get sick. I mean, like they were losing weight, so that means that the drug is attacking other cells too.
Okay.
Yeah, that's a great question. I don't know if it's at the same level as the chemotherapy, that is targeted agents.
Yeah, I'm sure like that's hard to tell this early on still to the research.
Oh, we just had a question come in about trametinib, and is that approved for kids or just adults?
No. Irenis is approved for kids too. Yeah, but basically you can ask to the doctor for this drug. Yes.
Yeah, so it's probably been approved for other indications.
Yeah.
And there's safety and dosing.
Yeah.
For trametinib. Great.
So are genetic alterations known for any of the samples? And if so, do the mutations trend with the chromatin clusters like TP53 mutant with cluster 1?
That's a great question. We do have the whole genome sequence for most of the samples, and we know that cluster 1 is most of the samples that are MYC amplified are cluster 1. For P53, most of the osteosarcoma tumors have a deletion in P53, so it's not cluster specific. But because we do have the whole genome sequence data, we can ask if these clusters are driven by copy number. And we did the analysis using only the cell lines, the PDX cell lines and the cell lines. And the answer is no to that. The clusters are not driven by copy number. But yeah, that was a great question, actually.
I'm curious if from the drug testing that you did and with the response, did you see did you see any kind of change? So in the cell states, in the clusters after, like, did it affect the clustering? Because, since it can kind of, it's a little bit plastic, can kind of move from one to the other.
Do you mean after treatment?
Yeah, exactly. Like in response to the drug, I'm just curious how that might impact the cell states.
Yeah, that's a great question. Actually, right now we are doing ATAC-seq libraries of these tumors after treatment. So we don't know the answer yet, but we will. That would be great, actually, that if you treat with one drug, then you can have most of the cells reverse or go back to the other cluster. Yes, that's the hypothesis, but we don't know yet.
There is a tremendous amount of work in these slides, Dr. Lopez Fuentes. Like, as we are going through each slide, I can only imagine, kind of how much time it took to get all the data in all of these slides. It's pretty tremendous and impressive. So thank you so much. Walker, did you have any other questions? And I don't see if anyone has any other questions in the Q&A. Feel free to pop them in. But
No, I can't think of any. The slides are very information filled, so that helped.
yeah, it's and it's great that you already kind of have your next steps teed up based on what you've learned to date. So we're excited to hear more. I know we're kind of we're definitely looking forward to seeing you at Factor and hearing more at that point too, about what you continue to learn on this study. And thank you for joining us on Osteobites and for making it better for osteosarcoma patients everywhere. And more information about this and all Osteobites can be found on our YouTube channel, also on our website at mibagents.org. And you can also listen at your favorite podcast place. We are going to be off for the next couple of weeks. We're actually going to be in Denver for a mingle and also to visit our 2022 Outsmarting recipients, Dr. Dan Regan at CSU and Dr. Michael Leibowitz at the University of Colorado. But we'll be back on Thursday, February 16th with Dr. Jayantha Parasarathi from Nationwide Children's Hospital. And she manages the 3D printing lab at Nationwide Children's Hospital, which provides comprehensive patient-specific treatment planning and device development services from a pediatric perspective. And she will discuss the virtual surgical planning and 3D printing in pediatric musculoskeletal oncological resection. So definitely sign up for that. Should be a really interesting one. The link is in the chat. And you can also visit our website to see the Osteobites lineup for the next few weeks. And if you have ideas for future topics that you'd like to hear about, please let us know. Just email us at events@mibagents.org. Thank you again, Dr. Lopez Fuentes and Walker, for spending an hour with us today. And thank everyone else for joining us on Osteobites. And we hope to see you in a couple weeks when we chat with Dr. Parthasarathi on February 16th. Thank you so much.